Hello, everyone. This is week number 3 for Genomics in the Cloud. I'm your host, KT Picard. Thanks so much for being with us here today. There are about 5 or 6 of us on the call, and today is chapter number 2, Genomics in a Nutshell. We'll have a discussion after that, and of course, a repeat of the resources that we discussed during the talk. I'd like to ask, we kind of hold questions. There's a lot of material in this chapter. It's one of the heavy lifting chapters because it covers so much. I'd like to ask to hold questions to the end. If you could, just jot them down. That'd be very helpful. But I'll also ask for the question if there are specific things in genomics that you have questions about. That we have to skip through a lot of material just because of the amount that there is out there. But if there are specific things that you'd like us to talk about, I'll pause here and see if I can work them in. I'll do my best. Okay, so there's a lot that we move through in the book quickly in chapter 2, Genomics in a Nutshell. We kind of eliminate a lot of the preliminaries around cell biology and specifically around the cell cycle. But what we come to after you've looked at the 23 chromosomes that make up the human genome, the autosome, the 22 chromosomes of the autosome, and the last chromosome being the sex chromosome, what you end up with is one of these things here, which is a chromosome 1 of 23. And this slide unpacks that then from chromosome all the way down to base. So for those folks who haven't seen too much about genomics, I thought I'd start here. And what you're looking at in the upper left is a chromosome, the one half of which is we refer to as a chromatid. It's important to remember that in this picture, these chromatids are called sister chromatids. It's the replication of them. They were getting ready to divide the cell. And most of the time, when you look at like a karyotype, for example, karyotyping is still often used here in the States to look for genetic abnormalities. It's the analog version of what we're about to embark on. And it's where you take the 23 chromosomes and stain them. You'll notice that the chromatids are separate from each other. And that's where they spend most of their lives is separately that way. But when you do get them ready to divide, they look like this. They're gathered in the center. It's something called the centromere, which makes sequencing of that region very difficult because of the way that they intertwine. You'll see the word telomere, meaning the ends of the chromatid, and there's a number of bases there that are unique that make it easy to spot when you're at the end. There's a short end and a long end. So the short end we call the P end, and the long end we call the Q end. So you'll see in references, when you refer to places in the genome, you'll see, for example, you might see chromosome 1, 2, P3, and that will refer to on chromosome 1, the second exome or exon in the chromosome, and then spot number 1. So let's pull that apart, because as you roll down the genome here, you'll see this being unraveled. Again, not enough time to get into histones and how all this stuff, is supercoiled and all that. But when you do take it apart, you end up with pieces called exons and introns. The exons are the portions that encode for proteins in the gene. They make up somewhere between 3 and 5% of the genome. The introns then are the things in between, formerly called junk DNA, but as we learned, they play a large role in gene expression and other areas we're just starting to learn about. I put some useful mnemonics up in the upper right. As we move down now to the bottom, you'll see the individual bases, the A, C's, G's, and T's. I use all of those genetic codes mnemonic because A's and T's bond together and G's and C's bond together. So when we talk about adenine, thymine, guanine, thymine, those bases, that's how they bind together. Again, time doesn't really permit us to get into how all that happens, but again, a useful way to remember them. So exons are exciting, and you can see their introns are the bits in between that might help remember the difference between the two. So now at the bottom are the nucleotides or bases that comprise DNA, diribo. It's been a while. Deoxyribo. You guys can look it up. A lot of these acronyms you can find on genome.gov. These things are described in wonderful detail. The human genome has about 3 billion base pairs inside of it. As I mentioned, in 23 chromosomes, they're in the nucleus of all of our cells. Finally, I'll conclude with when you wrap together, exons and introns, you get these things called genes, which are kind of squishy to define, but the book defines them roughly as a unit of inheritance. There are lots of definitions for this, and right now the count is somewhere around 30,000 genes in the human genome. Again, most of which are used in the production of protein, which leads us to our next slide about the central dogma of biology, famously written about by Watson and Crick in 1958. Watson kind of cheated it a little bit and said it was the movement of DNA to RNA, and then from RNA into proteins. Crick was a little more specific about it, and really his comment was, you can see at the bottom left, once information has passed into protein, it can't get out again. And so his point was, it's a one-way movement. As we've learned, there are special cases for this, and sometimes we go from DNA to protein, and there's other ways of replicating. There's special cases for all this, but it's seen in almost every biology textbook. This process of taking DNA and moving it to RNA in a process called transcription. The RNA then has the introns removed, so it has only the exons or the protein coding portions in place. Those then move into a process called translation, and that's where we take the RNA, which is the messenger that tells a complex, a ribosome, how to build a protein, which, as you can see, sometimes is folded into different shapes or isoforms, as we call them. Again, I oftentimes use the mnemonic, translation happens after transcription, and so you can see up there that translation is later, and that's how I remember the two. The process of translating the messenger RNA into amino acids is a remarkable process, and this table describes the method that we actually create these three amino acid pairs, often called codons, and these three amino acid pairs, also referred to as amino acids, are used then to string together to build into proteins. When you take 50 to roughly 2,000 of these together, you have proteins that are used in the human body. The median length of most proteins is about 375 amino acids. The amino acids themselves, or as I mentioned, codons, are these three or three pairs of or triads of bases. And an example you can see up on the top right, we'll walk through that. This is the DNA sequence, and you can see it's TAC, TTG, ATC. A small note is that we in the world of translation, T becomes U, and so it's uracil is closely related to T, and so that's the only difference letter-wise. And so as you can see, the complementary pairs along the bottom. So going from top to bottom, T becomes A becomes U, and C becomes G. And if you look at that sequence under RNA that's coded in green, AUG is called the start codon, and the start codon is the message that says, hey, start looking at what I have to say from this point forward. And as you can see in the table here, you look at the first letter A, the second letter is U, and then the third letter is over here G. So AUG is the start codon. We look at the second complementary sequence here, and it's AAC. And again, when you use the decoder table here, you see it's asparagine. And then the last one is UAG, which is over here, which is a stop codon. So the amino acid that we produced is asparagine. Again, if you string these together, anywhere between 50 and roughly 2,000 of them, you get the proteins that we use to make everything. So it's an amazing, it's an amazingly magical system. But again, in the time that we have, that's as far as we'll treat this the process of translation and the like. So moving on to mutations, this now gets more toward the bioinformatics space that we're interested in about, and is understanding how normal genes produce these proteins, or in the case where there are mutations where they do not. And depending on how things are mutated, you either get a protein that doesn't function as well, or a loss of function, or no protein at all. And this is really where we start to get into the movement of looking at sequencing and looking at the ACs, Ts, and Gs, and how they now map into the digital world of bioinformatics. The book goes into a fair amount of detail at this point about sequencing itself. It talks about Sanger sequencing, the beginnings of the sequencing process. It talks about, there's a section on Illumina sequencing and how that works. Again, in the interest of time, we won't be able to get into different methods of sequencing. But we jump right into what happens when you go from this analog form and start to break it apart. And that leads us to diagrams that look like this. If this looks new to you, it certainly won't by the time we finish the book club. And what this diagram is showing are different kinds of variation in the genome. Along the bottom, you'll see the different kinds. So we have short variants, in the middle copy variants, and on the right side, structural variants. So what you're looking at on the vertical axis on the left are pairs of reads, and they're what we call piled up, and again, the kind of variation. So the blue line across the top is showing that all of this is happening in this example in chromosome 1, and you can see that we're starting to read these things out and get a sense of what's going on. Again, from left to right, the easiest one to look at is something called the SNP, or single nucleotide polymorphism. This is a point mutation that occurs at a frequency of at least 1% in the population. So these SNPs occur about 1% of the time per individual genome, or about roughly 1,000 nucleotides on average, which means that each person has 4 to 5 million SNPs in your genome, which is why we're all kind of 99.9% similar. As I mentioned, there's about 3 billion base pairs. So that's what encodes the wide diversity of human phenotypes. We found more than 335 million of these SNPs across humans in multiple populations. When we talk about sequencing from companies, or often called genotyping, from companies like 23andMe and Ancestry and the like, those are what we're talking about, is looking at just those 4 or 5 million single nucleotide polymorphisms, or variations sometimes if they're in the somatic cell. Moving to the right, insertions and deletions, lovingly called indels, is the class of variation, and we'll talk a little bit in the next slide about all the different kinds. But these are inserts and deletes of various genetic information, and they're important for understanding changes in function. Then we have some different kinds of deletions moving to the right. Homozygous, meaning you've lost function in both alleles, or allele meaning the description of a gene at a particular location or locus. If you've lost half of it, you're so-called hemizygous, or heterozygous in that case. There's over to the right then, amplification, where there's extra copies of information that leads to different phenotypes. An example would be Huntington's disease, where there is a large number of excess alleles for a particular location on the gene, and that causes some serious issues. And then finally, structural variants, which are places where things really go crazy, and where chromosomes tend to hook up with other chromosomes in unexpected ways. We see this most often easily in cancer, but it happens also in some inherited defects. And we'll talk about how those things are sometimes inherited or sometimes just happen spontaneously. I mentioned structural variants. So these are all the kind of ways that you can move things along. Along the top line, you'll see the reference. So these are different genes, A, B, C, D. And I won't go through all of them, but I think it gives you a sense of the tremendous number of ways that you can rearrange these things, from inversions to deletions to, I love this bottom one, a complex, hairy thing. And I think that's the technical description when you can't bucketize the variation into one of those pieces. So lots of different ways that these things happen. At this point in the book, there's also discussion about different ways these happen. There's discussion about errors and other ways that these things are called incorrectly. Again, time doesn't really permit us to go into all the different ways that things creep in. What we can talk about is this thing of how things are inherited. Again, we skip through a lot of things like Mendelian inheritance and penetrance and all those just kind of things that are in general biology. But this slide is useful because it talks about things that happen in the germline, germ from to germinate a cell or a seed, versus a somatic alteration, which happens, generally speaking, after we're born. And it's a soma being from the word body. And the tricky bit here is that somatic alterations also occur while we're developing. So you'll see that orange little dot in the middle of the little blastocyst there. And it happens, normally these are things that are inherited from your parents. Again, we don't have time to get into, how the gametes are created, because there's meiosis here that we won't be able to cover. But it does talk through how we get to places that are homozygous or heterozygous, and how we get into those states. And whether we have one or two copies missing for a particular gene. And it, again, walks through the different parts or different aspects of life. I will point out that this is not orange leg syndrome is not actually a thing, but it is pointing out that errors occur during the replication process while we're being born. Also during life, there are ways that it could be external forces like radiation, or factors like the environment that have to do with epigenetics or changes in expression of genes. That occur over time. We also accumulate variation over time, or mutations over time. Men, I know, at the rate of about 2 mutations per year as the cell divides. And these mutations can then be passed on to our offspring, which is at the bottom, which again, we don't have to get into, we don't have time to get into meiosis or the production of gametes, which are then used to propagate the species. So again, a very quick tour of genomics. That's more or less a semester's worth of genetics in about, I don't know, 15 minutes or so. What we go to now is the fun part and why we're here. We'll talk now about the bioinformatics part of this, and now we're actually talking about where the ACs, Ts, and Gs end up. And this is the translation of the analog into the world of the digital. We began our tour with something called the FASTQ file format. FAST, I don't think really stands for much. Q is for quality. Its predecessor was called FASTA, and it handled, the A was for all types of data. It was introduced back in 1985. And you can see here the record and its various components on the left. It has a sequence name that has some standard coding. And there's the nucleotide sequences themselves. And that sequence, you can see CT, maybe a little difficult to see, but anyway, it has the bases there. And then at the bottom is a score called a Phred score, Phred. which has no meaning at all other than there was a program once upon a time called FRED, and it's a logarithmic scale that's used to show the quality of the score and how confident you are that that base is actually the one that you think it is. You can see on the right then what we're looking for are FRED scores that are generally higher than 20. Oftentimes we just discard data that have lower FRED scores. Most importantly, FASTQ files are for humans, about 200 gigabytes uncompressed and about 60 gigabytes compressed. This varies a little bit, but it gives you a sense of why we have this explosion in genomics. And when we talked last time about the 1 to 5 million or so human genomes that have been sequenced, you can see that we quickly get into the petabyte and beyond exabyte range with data that there's a huge explosion. And this is kind of the first place where it starts. These FASTQ files, again, we don't really have time to get into it, but they're generated on sequencing machines. The paired end reading that comes out of the Illumina technology, the original Solexa technology, is where most of the time we spend in the book. And they talk about how you can use paired end reading for additional accuracy. But suffice to say, these are generally around 200 base pair segments that the sequencers produce. You may have heard of long sequencing, long read sequencing from PacBio, for example, and those go into much in the thousands, 10,000 lengths of base pairs in one shot. You still have to align them into so that they line up. One of the things we talk about is how many of these things we put together to actually get to figuring out what is really there. And so that leads us to mapping those segments to a reference. I mean, really the whole process of sequencing is about this jigsaw puzzle, which is these small 200 base pair segments, and then putting them together and finding where they overlap, and then moving through them this way vertically to make a call. And so the first process that we go through is changing the FASTQ file, looking at a reference genome and mapping them to, first thing is called a sequence alignment map, or SAM file. Its binary equivalent is called the BAM file, and that's one you may have heard of. The BAM file is the binary equivalent of the ASCII text SAM file. And so it's smaller. It's generally, 4 to 5 times smaller than a BAM file. And again, as you can see on the bottom, there's some of the acronyms, the sequence alignment map, the BAM at about 80 gigabytes uncompressed, and a more recent file format called CRAM. CRAM doesn't stand for anything other than it's a play on cramming data into a smaller space. It's a compressed BAM file, and it's generally around 50% smaller than a BAM file. So it's a newer technology that's being used to save on space even more so, but it does require compression against a reference genome. The first one, which was published about 20 years ago in the Human Genome Project that we talked about before. So here's the quick tour on the BAM file. It has a header that you can see has a dictionary and some things called a read group. And then within that, there's one read per record or line. And you can see the various parts here. Again, it's something that we'll become more and more familiar with as we go through the book. Some things to point out from left to right there on that line. Again, the read name. I'll move to the position. You can see CHR referring to chromosome number 1. And then again, how many nucleotides you've moved on the short peak or long Q end of the genome. I'll move across to CIGAR, which is the gap alignment, which is explaining a little bit more about how the read, maps. Is it an insertion? Is it a deletion? Is it valid for a number of nucleotides? The mate information is used to pair up with other sequencing similar reads as we talked about. The read sequence is the raw ACs, Ts, and Gs. FRED is next, so you have quality scores there. And then some metadata or some just extra flags that help you understand what it is you're looking at. Again, lots to go into here. I'll provide a link at the end for getting at this, but again, we're getting at the code of life and how we actually put it into practice and share it with others. Just a word here on whole genome versus exome sequencing. Now that you've seen these pileups and how they line up, you saw the earlier slide where we talked about exons and introns. When you sequence only the exons, you get an exome, which you'll see at the bottom here, which doesn't include the introns or intergenic regions of the genome. Up along the top, you'll see whole genome sequencing, and you'll notice then the check marks, the green check marks and the red X's. And the difference there is that we're sequencing on top all 3 billion base pairs in whole genome. On the bottom, you're sequencing only the 3 to 5% of the genome that is responsible for encoding protein. Now, again, that's where a lot of the action occurs, but not all. And what we've learned is there's a lot of things that really we can only find out using whole genome sequencing. The reason that we haven't all just jumped to it right away is because, of course, cost, but that's rapidly declining. And I'm hoping we have some time at the end to talk about a paper that was published this week that purports to move the cost of whole genome sequencing down to even $15 US, which was pretty exciting in the last week. Okay, so the last stop on the bioinformatics tour, we talked about FASTQ, we talked about BAM files, and the last one is called VCF, or the variant call format. So this is when we make a decision about, I'm going back to the previous slide, we look at all these different, the coverage here. The number of times this occurs is called the read depth, and the read depth is the number of times or the number of copies we have of an ACT or G at that particular location. We use programs like GITC to make a decision about which way to call it. We gather or aggregate all those calls in something called the VCF file. And as you can see, it's the smallest file that we have, and it's at 2.5 gigabytes compressed. So the quick one on that is, again, it has a header. It talks about, what's in there. It's, again, you can see it there, the depth and the quality scores and how things are coded. And then the records themselves, you can see some of the same kinds of information, the chromosome, the number. This is a SNP ID, an RS number that you can type into a number of databases to get information about that particular location. Where we are on the reference and what would be expected there, or an alternative for that particular spot. I get a quality score. Did it pass your quality filter? The read depth, or how many occurrences of that occurred. The allele frequency, again, which we don't have time to get into. And then a little bit about the format of the data that you're about to see. Then we have, you can put in multiple samples into a VCF file. So this NA1, NA2, NA3 refer to 3 well-characterized genomes maintained by something called the Genome in a Bottle. It's a consortium of kind of standard genomes that are well-characterized. So again, without getting into too much detail here, you'll see that the depth in this particular location, there were 14 reads. There were 8 reads and for this one, and 5 reads for that one. So the 1-1 is how we're calling it in terms of, is it the same as the reference? So is it homozygous, or is it different? Is it heterozygous? Is one copy the same as the reference and the other not? These things will become much more clear as we start to look at real-world examples inside of a genome browser, where we actually see the pileups of A's, G's, and T's. So that's a quick tour. We talked through some number of file formats. Again, the binary world of bioinformatics. Again, those 3 file formats. I should add one for VCF. I'll add that. It's important that these things come together the same way, the same time, every time. So there is a consortium that's mentioned in the book, a paper referenced here about building a functionally equivalent pipeline so that we can exchange information between facilities without worry, and they all come out the same. Finally, I mentioned the reference genome. There are a number of them out there. The two most common are the older one called HG19, and for reasons that we don't have to get into, the next one is called HG38, has a number of different synonyms for it. For example, you can see GRCH38, but this link talks about why those are different and the additional information that is in HG38. And it's important that when you pick a reference genome, that you stick with it. So as we get through the book, we'll be referring to these more. Again, not enough time to get into even things like graph genomics. All the things we'll be talking about are capturing variation in a linear fashion using the tools that we mentioned in last week's talk. So how am I doing on time? Looks pretty good. This is where I wanted to be, so I'll pause here. Of course, next week will be on chapter 3. We'll talk about computing kind of in the same way, so that if you're a genomicist, but computing is new to you, we'll do the same kind of whirlwind tour in a nutshell. But today was genomics in a nutshell. I hope you found that useful if that's a new field for you. But let me stop sharing and we'll take time for questions and go from there. So everyone's
so I have a question.
Sure.
So it's actually about building reference genome. So I think, as you all know, that our reference genome is very biased for the Western populations, because mostly that's where all the science or research were done. But then, for example, if you want to build a reference genome for another population, then what would you do about that? And my question would be, how many people, so when you're building a reference genome, how many genomes would you have to sequence until you can confidently call that, okay, this is the reference for this population? Because
right. Yeah, thanks for that question. The original, it is a challenge, and it's one of the things that we talked about. If you missed it on Slack, there's a nice discussion about sequencing for populations that are not well represented in the reference genome. I think originally, I think it's an amalgam of 5 genomes, heavily influenced by Craig Venter. His genome, of course, is mixed in the reference. For various populations, it doesn't map terribly well. So there's a lot of variation that can be captured. The newer reference 38 tries to get at that variation, but eventually we're going to a place of de novo sequencing, or sequencing from no reference, right? I mean, it's kind of the ultimate goal for all of us. Computationally, it's still really challenging. So and for compression reasons, we often, call against our reference. But you make a good point that this doesn't capture all the variation that's out there, and it is limited to what we put into the reference. I know there are people on the call that actually deal with this on a daily basis. So I'd love to get input from others about how they've dealt with variations in their populations, whether they're founder populations, small groups of people that are genetically similar, or different, different subgroups across the world. So I'll pause there. I'd love people's input here.
Hi, I guess I could give a little bit of input back on that. I shared a bit in the Slack channel. If anyone wants to catch up, there was a kind of a paper out that's trying to merge the gap between these graph-based genomes, which we won't go into, in the linear genomes, basically trying to capture some of the some things that's unique to populations. So there's 2 studies. One was looking at an Icelandic population, and another was looking at around 910 Africans. And some were more conservative than others, but they found, there's large sections of the genome that are missed in these populations. So essentially what we're trying to do in Canada, and there's a similar thing going on as well with the Territory in New Zealand is we're trying to build a background variant library for the Indigenous population to better represent them, because there's just many sequences, like insertions, for example, are missed with these variant callers. So when we're looking at rare disease patients, there's this typically 50% of them with whole genome sequencing are still unsolved cases. And it could be that, you know, these are areas that just aren't captured in the current reference genome today.
Matthew, thank you. I was hoping you'd jump in on this because that's one of the topics, thank you, that you discussed in some detail on Slack. And it was really informative for me. So your input, as well as the input from the rest of the members in the team, it's made a tremendous, for me already, a tremendous difference in learning. So I want to thank everybody for their contributions. And I made a post a couple days ago about the kinds of things that have been discussed in the last week. I'm just kind of looking for it. That was one of them, was around genome diversity, which you just mentioned. There was a whole other section on workflow language selection. We'll get into that later on WDL and different approaches to that. There were discussions on reproducibility and some tips there. Even a connection on my end from connecting to the UC Santa Cruz database in R and how to do that. So we put in some little tips. So thank you for that. I appreciate the comment. Other questions?
Sorry, I'm just going to extend that a little bit more as far as like the references go. So there's the HG19 and then there's the HG38. And with variant callers, so with HG38, there's a lot of these alternative contigs, and the variant callers are not really designed to even look at these. In clinical practice, we're still using HG19, a lot of these tools that we're using. So for SNPs and stuff, they've been taken well care of. These big consortiums are now addressing structural variants in more detail. And so Nomad SV, they're still built on HG19, so they haven't caught up in HG38 yet. So we're still not even using that. So there's still lots to catching up to do. There's also issues like where you get it from. Do you get it from GenCode? Or are you getting it from Ensemble? And there's differences with annotations between those. So it's a really complicated topic. Anyone who wants to read more, I'll probably share a thing. Heng Li is talking about choice of reference genome for human populations. So I'll share that later and we can continue the discussion.
Oh, yeah. That would be great, yeah. And well, one more question then. So imagine if you're conducting a multinational cohort, maybe linking your variants to your to a certain disease. Then how would you actually align your the genome of your samples? Would you have like one reference genome in which the genome from all samples, from all populations, from all nations will be aligned to this genome, or would you have like separate genomes? Okay, so this is the reference for the Western population, and this is the reference for the Eastern population, and so on. Do you get what I'm asking?
Open that. Yeah, I do. I'm going to open that up to the group. There are probably people on the call who have a lot more experience than I do. Generally speaking, you know, we tend to align things to a common reference for convenience. And as Matthew mentioned, a lot of the work still is on HG19. Some labs in the United States have recently moved to HG38, which again captures more variation. But to your point, what happens when you have a population that's different? What do you do? I'd love to open that up to the group and ask what people have done.
Oh, this is Adelaide. Hey, I was just posting about Nomad.
Yeah.
Which is an international consortium trying to harmonize exome and genome sequencing to allow for biological insight across populations. So I'm sorry if you already went over this material, but I just thought I'd just pop that in there for because it comes up later, you know, in the book and in the resources, and it's a very useful effort, but it takes a lot of people to make this happen. So it has to bring together diverse data sets with different types of metadata. That's like one of the issues, you know, the metadata, how it was collected, who collected it, what database it was in. So a lot of this stuff is already being generated for researchers to be able to use this data. And this is another effort from the Broad Institute, you know, kind of generating all this good stuff for people. But I just thought I'd put the link there since it came up in the conversation.
No, that's great. Thank you, Adelaide. So Genomat, if you're not familiar with it, is fairly widely used now to look at allele frequencies in subpopulations. Adelaide, I want to say there's at least half a dozen that are provided there, maybe more now, something like that.
Yes,
Oh,
it's,
good.
it's a curated resource, and they're kind of updating, you know, as the human genome gets updated, they try to update the resource to match the more recent versions of the human genome. Yeah. Great.
So there's alternate variation that's captured there. Thanks to everyone for posting in the chat. I'll make sure that these links get pulled into Slack so that if you want to come back to it later, it's easy to get to. So appreciate that. Mikhail, did we get to your question?
Yeah, definitely.
I can add my answer to that question. So kind of bouncing on what Adelaide said. So you would essentially, if you had a lot of samples, you would align them or map them to a single reference. But of course, that reference, even putting diversity aside, that reference is only going to be one single haplotype. You don't, because of the fact that it's just basically one line of text. So it's always going to be a little bit inadequate to capture the diversity of the entire human species. So that's why things like Nomad exist. So you do your mapping originally, and then to basically either filter for or filter out the rest of the diversity that, for example, if you're looking at rare disease, you would tend to ignore. You use these databases like Nomad or what's another one? I was going to say HapMap, but that one's a bit old. DBSNP. There's a number of databases that you can use. 1,000 genomes. Yeah. There's a number of databases you can use to kind of capture additional diversity that you either want to eliminate because it's too common or that you want to focus on. So essentially, you can think of the mapping is just the reassembling together at the beginning, and then if you want to further kind of take into account the different groups of populations that you have, then that's where things like Nomad would come into play.
I'm wondering if anyone has experience. I'm thinking about this as well in the move. Someone mentioned earlier that most of it's on HT19 and HT38 is newer and captures more variation, but not everyone's moved to it. Rather than reprocess everything and recall all your genomes, I've recently seen, maybe in the last few months, software that's kind of lift and shift. So you can move your VCF file from HT19 to HT38 without having to reprocess everything. Has anyone bumped into that yet, or is it still kind of early days for these things?
Well, that's an incomplete answer to lift and shift. You can't really do variant calling, and when we get into the GATK stuff, you'll see why. Yeah. It has a lot to do with how the variant callers' underlying algorithms are written. So yes, you can have concordance. I guess you can look at things that were called by HD19, but it's actually just a completely different animal, the 38. So just throwing in my 2 cents there, having been someone who worked on the GATK message boards, and maybe Geraldine will comment later about this issue. But in a way, there's not a lot of ways to just do a lift and shift and have it be cleanly done and representative, because any variant that wasn't in 19 but now is in 38 is not going to be represented. And so that someone with like rare diseases like MET2, like you would, I don't know, do you have an opinion on that?
I do have an opinion. When I looked at the copy number variants, I tried to do the liftover, and it caused too many errors, and I said, okay, I'm just going to stick with HG19 for that data set. So I'm guilty of, I'm one of those people that hasn't properly moved on.
Interesting, very interesting. Okay, well, yeah, like I said, it's early days, and I hope the book does discuss it. I haven't gotten that far yet in the book, so yeah, that's really good. Okay, that was really an interesting conversation. Thanks to everyone who, there's a lot of good stuff in the chat, so I'll again make sure it's that's published. Good. Other questions? This is exactly what I was hoping for from this group. I mean, it's, obviously there's a lot of knowledge about bioinformatics in the group. So if this is new to you, this is probably completely overwhelming. And all we can say to that is it gets easier with time. I've been at this for about 10 years, and I have to tell you, when I read chapter 2, it was somewhat encouraging to me, because for the first time in my life, I realized I understand everything in this chapter. And that's actually a really good feeling. And I think for many of you who are teachers, there's no better way to learn information or to know that you know information than to have to teach it. And so I did want to thank you for listening to me talk about kind of a whirlwind tour, because it gave me a really good chance to crystallize a lot of ideas that have been floating around in my head for a long time. Other questions or thoughts there on things in the last week, questions about what we talked about, or the like. I'll pause one more time. Okay, well, we'll do a quick shift then. We've got a few more minutes here. I wanted to talk about some papers that came across in the last week, two of which I'll mention. One was the sequencing of a bunch of samples. I put it under sequencing palooza under the Slack channel. You can see the paper there. It's again from this Genome in a Bottle consortium, where they're looking at a number of reference genomes, reference samples, would be better said, and then with different variants in depth or read quality and different sequencers. So I think there's 3 Illumina sequencers and a PacBio sequencer. And you can look at this data. I posted the link on Google Cloud. You don't need a Google Cloud platform account to look at this data, but it's a tremendous resource to look at examples of the files we talked about today. If you've never seen these before, it's a great way to take a look. So there again, there's 2 trios in that, a mother, father, and a kid, and some other ones. And so just a tremendous resource for just base, again, well-characterized bases and calling for all the file types we just mentioned today. So that was paper number 1. The second thing was, in the last few days, there was a paper from BGI, which was kind of an extension of a previous result that they reported earlier this year, which was changing their kind of VAT or batch approach to sequencing, and changing it to one that was placed on putting reagents on film. And so this is using, PET plastic, the kind of things that soda bottles are made of, and separating these essentially creating a long roll and doing sequencing at very high volume, very high numbers of base pairs, and which would they claim in the paper that it would bring the cost of sequencing down to about $15 per human genome, which of course changes the game entirely. Someone pointed out it costs now more to ship the sample than it would to actually sequence the sample. So there are all kinds of economies of scale that could come into play there. But it is kind of interesting in the sense that we've been stuck at $1,000 for a couple of years now, maybe 3, and everyone's been waiting for this to happen. Again, earlier this year, BGI announced that they could, with the batch tank processing, get it down to about $100, and then with this one now calls it down to 15. So that's again, it remains to be seen. But within this year, we're starting to see claims made in papers that move the cost of sequencing down to a price where you could just basically sequence everything and multiple times at that. So I don't know if anyone's seen those papers, but again, I posted that the link to that paper inside of Slack. So I thought that was interesting. Any comments there or papers you've read in the last week or so that were interesting to you, or work that you've done that's been kind of fun?
Well, this is Adelaide, and I just moved over to the NCBI, so I'm working on trying to understand the new hierarchy and everything. And there's a lot of data mining to be done in the sequence read archives that is really fascinating. And I'm finding out a lot about how they're doing the virus databases, the brand new COVID, databases that came up since the beginning of the year, how far we've come. It's really amazing work, and the type of science that they're trying to apply to it is just phenomenal. And so it's so exciting right now, even though it's a depressing time because of pandemic, it's like actually a very exciting time to be a scientist in this field, because it's pushing our ability to get things done quickly. It's really bringing a highlight to the need for these larger shared resources, right? We're not going to get to the answer all by ourselves. So I just find that so fascinating. So just a little positive note to end the year on, I guess. Yeah.
Yeah, no, thank you for that. And it's true. There's an incredible amount of science that's going on really, really fast worldwide. And, again, our members today, again, are from around the world. And I'm sure many of you are working on these problems. The sequence read archive that you mentioned, Adelaide, is, again, a wonderful resource. I used it once to pull down a genome of green peas, Pisum sativum, to look at, what was the sequence that made yellow peas yellow and green peas green. And you can actually go in there and find the spot that got Mendel all excited, over 100 years ago. So a tremendous resource for all kinds of species. Also, yeah, a little bit tongue in cheek, but someone on Twitter this week said we can get from virus to vaccine in 9 months. And of course, that's faster than it takes to get a paper published. So I don't know if that's a comment on the academic process or peer review, but I thought it was a salient observation because for those of you who do write papers, you know what a frustrating process that can be. So in any event, it is an incredible feat of science. We oftentimes talk about moonshots and sometimes mischaracterize or use the word kind of inappropriately. And I think this one ended up being in that category, or Manhattan Project, or pick your country's favorite, Mission Impossible. And I think that's what we've been able to achieve, getting to a vaccine. So thanks for that, Adelaide.
I think that's a good point, Katie. I posted a link for the Nomad papers, and that was work that came out of the MacArthur lab in Broad. I think it was like 7 years. They went from Exact to Nomad, and the papers came out from preprint to publication. I think it was a year. And typically for Nature, it's like 2 years in the review process. So yeah, it's pretty amazing.
Right. So it's changing, right? And so bioRxiv, these preprint servers, this was maybe 20 years ago now, the physics community with arXiv kind of pioneered this approach. And there are now other mechanisms, kind of socially generated mechanisms for peer review. Time won't really permit us to get into all the changes that are going on in academic publishing, but suffice it to say that it's undergoing a tremendous amount of change. And if you haven't followed the open access movement, it's made for me a tremendous, I can't even tell you how much I've learned through open access papers. Because I'm not an academic, I don't have access normally to papers. I'll make a shout out to Sci-Hub if you haven't seen it before, sci-hub.se. And I may burn for it, but that's a site that has provided me with a wealth of information over the years. And it is definitely in the crosshairs of a number of publishers, but it's, again, something that has been tremendously useful for getting at source information and understanding the latest changes that I see going on in genomics.
We'll have a chance to talk about, sorry.
Go ahead.
I was just saying,
won't be publishing on it, but yeah, go ahead.
We'll probably have we'll have a chance to talk about preprints in chapter 14.
Indeed, yeah. There's a whole chapter on yes, and you'll be leading that. That's great. So chapter 14 on reproducibility. We had a nice discussion on Slack about that, getting at the beginnings, and Adelaide made some nice comments about how she does that. So I encourage you, if you haven't gone through those threads, to take a look. Some really good contributions from our members. Well, great. We're almost at time. I'm just taking a look at the chat. I'll summarize this and again post these so that everyone who is not watching video can see them. There's a interesting debates that we could have about on all kinds of things, from I don't know, even on reference genomes themselves. So I really appreciate the comments. I'll end with there was a post about human genome sequencing and having your own done. It's becoming more and more affordable. I summarized a few of the more commonly used ones here in the States, and that's there in Slack. Someone also mentioned a link to Mendel's Pod, which is a podcast that I can't say enough about. It's really how I learned everything that I know about genomics. I listened to every single episode of that show when it first came out. While I was doing the dishes, that's my, that was my learning, was that show. So can't say enough good about mendelspod.com.
I'll second that.
The mention was, yeah, excellent. I'm sure many of you that, big followers there. There's a discussion, a mention in the Slack channel about whole genome sequencing. And the show does often talk about topics like direct-to-consumer and labs that are doing sequencing. So if you're looking to get your own data sequenced, take a look at Slack, take a look at Mendel's Pod for additional information. Good. So final comments before we go off to our respective weeks. Was this how is the level of detail in the genomics discussion? Was it like too much or about right, or I already knew this stuff? I'm kind of just got a quick poll. Give me a hand raise if you would. So if that was like too much detail, Adelaide, I can't see you, but I can't imagine that was too much for you. Anybody too much? How about not enough? I mean, we had 30 minutes. Okay, so I'm going to assume that it was just right for everybody. We'll go with that. Any other comments before we close it up?
I wish I had that chapter when I was doing my PhD. Oh,
well, yeah. I mean, kudos to Geraldine and Brian. It is a nice summary. Thank you, Adelaide, for the thumbs up. It is a nice summary. Again, it omits a lot of things. This is such a vast topic. But I think in those short number of pages that are there, it manages to cover the whole process of getting from gene all the way down into bioinformatics and into the brass tacks of, into the details of how it is that we'll get our work done. So with that, we'll wrap it up. We'll talk again next week. A similar kind of quick tour on computing, everything you wanted to know about computing, but were afraid to ask. It's more on the computational side and less on genomics. Those will wrap up our kind of primer chapters. We'll get into the Google Cloud Platform after that. And again, I encourage you to take a look at GCP if you haven't yet. Start getting your account set up so that we can delve into the best practices in GATK workflows. So thanks everyone for attending. Again, really appreciate the turnout. Everybody from around the world, have a great week, and thanks for being here. We'll see you next week.
Thank you.
Bye-bye, everybody.